Hello, and welcome to our Unit 4 Genetics Concept 1 notes on DNA structure and replication. Okay, so before you start taking notes, let's just talk a little bit about DNA. What is it? So DNA is just a molecule that contains our genetic information. Just plain, simple, sweet, simple, and to the point. Our DNA is passed down from parent to offspring, and we get it from our parents. And this DNA, this molecule, these instructions, this genetic information is passed down from our parents and determines the color of our eyes, the shape of our nose, which is pretty important. Basically, everything about you is encoded for in your DNA. And DNA can be found, as always, within the nucleus of every single cell in the body. Okay, so it's a very important molecule. And then really think back to why it would be advantageous for DNA to be located within the nucleus, and think about that analogy that I mentioned last semester of how it's important to keep the things that are very important to our survival well protected, kind of like how our hearts are literally keep us alive by pumping blood through our veins and our arteries. And our bodies naturally have over time evolved through the process of natural selection to favor individuals or organisms that strongly protect their heart by having a rib cage. So all of those ribs that protect your heart, think of that as analogous to your DNA being located within the nucleus of your cells. So just an added extra layer of protection. Okay, so the notes begin. Nucleic acids. So nucleic acids are macromolecules that carry our genetic information, and DNA is just one of those molecules. And so DNA contains genes, or the blueprints or the instructions for making proteins. And these genes are located at certain points in a chromosome. Just gonna try like that. There we go. Okay. And so these proteins are very important because proteins carry out all cellular activity. They act as hormones. So any hormone you can think of, I'm not sure, I'm trying to think of one that maybe you guys would know. Insulin, for example. Insulin is a hormone. Insulin is a protein. Proteins act as enzymes by catalyzing or speeding up chemical reactions in our bodies. If we can recall from Cracking the Code of Life, little Hayden, the 2-year-old boy who had Tay-Sachs, the gene that coded for this protein that dissolves fat in the brain, so that was that protein's job, he had a mutation within his DNA located in that specific gene on that specific chromosome. His DNA was mutated, and so that protein was never made. It was never coded for correctly. So as a result, pressure built up in his brain, and this eventually, led to his death, obviously, which is really sad. But it was just one tiny misnomer, one tiny mismatch that led to that one protein not being correctly created. And so, yeah, proteins carry out all of our cellular activity. They're very important. So nucleic acids, there are two types of nucleic acids that we're going to talk about. The first one you all know and love, hopefully at this point, or you hate. First one is DNA, which stands for deoxyribonucleic acid. Deoxyribonucleic acid. Okay, and then the second type is called RNA, and the full name is called ribonucleic acid. And the only difference between these two are the type of sugar that is composes the molecule. So in deoxyribonucleic acid, DNA, the sugar is deoxyribose, and the sugar in RNA is ribose, which we have already been introduced to. So yeah, that's the only reason. Okay, back to the structure of a nucleotide, which hopefully we are experts on at this point, but if not, so nucleotides are the monomer that make up the polymer that is a nucleic acid. So nucleotides are really tiny molecules, and when you hook up a bunch or you bond a bunch together, a bunch of these nucleotides together, you build a nucleic acid. So a nucleic acid is a big molecule. Like DNA is a big molecule. And so that's what I want you to think of in your brain. Nucleotides are really small, and they have 3 little parts, and then a bunch of those together build a nucleic acid. So there are 3 parts. A sugar And in DNA, that sugar is deoxyribose, and in RNA, that sugar is ribose. The second part is a phosphate group, as you can see here, like we've seen before, an ATP. And then the third part is a nitrogen base, as indicated over here. And there are 5 different options of nitrogenous nitrogen bases if we're talking about nucleotides. So if we're talking about a DNA molecule, then the nitrogen base, so the sugar would be deoxyribose, the phosphate would be, it's just always a phosphate, phosphate's a phosphate, and then the nitrogen base could either be adenine, guanine, or cytosine, or also thymine, if we're talking about DNA. Adenine, guanine, cytosine, or thymine. However, if we're talking about RNA, so the sugar that would be located here would be a ribose sugar, and you don't really need to know the difference. It's just the difference between there's one extra atom or one less atom between the two different sugars. So for RNA, the sugar would be ribose, phosphate would be a phosphate, and the nitrogen base could either be an adenine, a guanine, a cytosine, or a uracil. So uracil is only found in RNA, and thymine is only found in DNA. Okay, so the structure of DNA. I know you've all seen this image before, this ladder-looking structure. So we like to say that DNA is structured in a double helix structure. So basically like a twisted ladder. And so the sugar and the phosphate that we just learned, or make up DNA, or make up a nucleotide that form nucleic acids, once you bond a bunch of them together, the sugar and the phosphate form this what's called the sugar-phosphate backbone. So you can see this here. So you can see here's the phosphate and then the sugar, phosphate, sugar, phosphate, sugar. And so this is known as the sugar-phosphate backbone. And what's happening in the middle is these nitrogen bases, so you can see these complementary base pairs, are actually bonded together with weak hydrogen bonds. So these bonds here are a lot more fragile than bonds in the rest of the molecule, because all of the other bonds are strong covalent bonds. So the bond between the phosphate and the sugar is strong. The bond between this sugar and this phosphate is strong. All these bonds are much harder to break than the bonds between those nitrogen bases. Okay. So continuing on, so if we were to kind of untwist that DNA, it would look something like this. So the nitrogen bases bond only to what we call their complementary base pair. And again, remember, they bond together with these weak hydrogen bonds, so very weak bonds. And A's always bond with T's, adenine bonds with thymine, and the C's always bond with the G's. So cytosine always bonds with guanine, and you can see this here. So if we're looking just at this strand over here, C always binds with G, A with T, C and G, T and A, G and C, T and A. So they always bond with each other, and we call these the complementary base pairing rules. So a cytosine, a C, is never going to bind with an A. A T is never going to bond with a G, and so on and so forth. They only bond with their complementary base pair per the complementary base pairing rules. Okay, so let's talk a little bit about what RNA is. So I mentioned it earlier. That is our second type of nucleic acid that we're going to be discussing. So DNA is a double-stranded molecule, whereas RNA is just a single-stranded molecule. So there is a single strand of nucleotides, and their bases are exposed. So they do not have a complementary base pair. So you can see here, we're only seeing one half of the puzzle. So think of it as like half a DNA molecule. Okay. However, so RNA bases actually bind with DNA bases, and we're going to get into what I mean by that in just a second. But the big difference that you need to know between DNA and RNA, like mentioned earlier, is that when we're talking about a strand of RNA, thymine is not present. Instead, anytime let's say an adenine needs a complementary base pair in an RNA molecule, that RNA would bind with that exposed DNA, half of that DNA molecule, and uracil would just basically think of uracil as a replacement for thymine. Did I make these rules? No, it just kind of happened that way. But just know, in an RNA molecule, there is no thymine, and if thymine per se is needed because we need to find a complementary base for adenine, we would go ahead and use uracil. So this is just here. So A's bind with U's and C's bind with G's. So try this. See if you can figure it out. So the types of nitrogen bases, so remember, are A's, C's, T's, G's, U's. Which ones are DNA, which ones are used in DNA, and which ones are used in RNA? Now, what about the type of sugar used? So in DNA, the type of sugar, oops, sorry, I forgot to give you the answer. So yeah, if you put A, T, C, and G here, then nice job. And if you put A, U, C, and G on this side, nice job. Okay, so back to the sugar. So DNA, the sugar used. RNA, the sugar used. If you said deoxyribose and ribose, congrats, you just got it right. Nailed it. And lastly, the shape of DNA. What did we say the shape was? And then what about RNA? What did we say the shape was? Awesome. If you put double helix or double stranded and then single stranded for RNA, you just nailed it. Okay, so really quickly, let's just go over some additional vocabulary that we've talked about before, some basics of heredity. So chromosomes, when I want you to think about a chromosome, I just want you to think about a tightly coiled strand of DNA. So different organisms have different numbers of chromosomes. So for example, like I said, humans have 23 pairs, 46 total, and you get 23 from mom and 23 from dad. So so when it says 23 pairs, so you know that typical image that you see. So this would be 2 chromosomes, or they would be homologous chromosomes. So you get this one from mom, this one from dad. They have the same genes located in the same places, and they exist together. And in every single cell in your body, within the nucleus of every cell in your body, you have 23 pairs of these chromosomes that are just tightly coiled strands of DNA. Okay. However, for example, like dogs actually have 37 pairs. So in every cell in their body, they have 37 pairs, or 74 total. So when we're talking about what a gene is, so now we know chromosomes, what about a gene? So a gene is just a specific piece of that DNA that has instructions to code for one protein. Okay, so it's just that DNA, like I said, nucleic acids are big molecules. So remember, we have 3 billion base pairs of DNA located in our bodies, and so that is a long, long strand. So a gene is just a very specific cut of that long strand that codes for a single protein. So why this is important is because you make sure you know one chromosome can contain thousands of genes all linked together. So let's go ahead. I think there's one more thing. So genes are pieces, sections of DNA. Hopefully I've slammed that into you guys enough. And chromosomes are just long strands of DNA. So think if we stretched it all out and then we bunch it all up, that we get that coiled look, that chromosome structure. And so this is what I was just explaining. So here, at a very minute, small level, we can see our A's and our T's, our G's and our C's binding together. We got our sugar phosphate backbone with our nitrogenous bases, hydrogen bonded in the middle, covalent bonds in throughout the rest of the molecule. And then DNA, we can see that it coils and coils and coils and coils and coils until it starts getting super tight together. And then by the time we compose these chromosomes, you can see how bunched up they are. They're super small, super bunched up. And then if we were to zoom out even further, remember we have 46 chromosomes, or 23 pairs of chromosomes within the nucleus of every single cell in the body. Whew. Got it? It's okay. If you didn't, just go back and rewatch.